This is a production of Cornell University.
Hello, my name is Larry Smart. I'm a professor in the Horticulture section of the School of Integrative Plant Science, and I'm based at Cornell AgriTech in Geneva. I've been at Cornell for 12 years now. Previously, I was a professor at the SUNY College of Environmental Science and Forestry in Syracuse, where I started studying shrub willow as a bioenergy crop. So let's talk a little bit about some of the urgent needs in society that we can address with agriculture. The most urgent is to address climate change and to reduce the amount of carbon and other greenhouse gases in the atmosphere. Shrub willow can address that issue because it can be used as a source of liquid biofuels, which, to be honest, are being replaced by electric vehicles at this point. It can be used as a source of electric power by burning wood chips in power plants, which, again, as we switch to electrification of our automobiles, can serve as a carbon neutral fuel source for electrification. We can also use wood chips as a source of heating fuel, and we do that here at Cornell AgriTech using willow that we grow on our farms, burned in a boiler that heats two of our buildings just a mile away from where the willow was grown. So local energy. We're also looking at willow for other ecosystem services. Willow can be planted in riparian buffer zones to take up nutrients and protect the water quality in our lakes and watersheds. We're also looking at a dual purpose for willow in that respect, in harvesting it or growing it as a fodder crop for small ruminants, sheep and goats. And we see increasing production of sheep and goats in New York right now. So those are the end uses. Again, right now, a push for biofuels in our breeding program inspired us to look at biomass composition. And we did a lot of work to quantify the cellulose, lignin, and hemicellulose in willow to try to optimize it for conversion to biofuels. That era seems to be fading away. So really our main objective in bioenergy crop breeding right now is yield and sustainable yield, but also carbon sequestration. So can we also identify willows that sequester carbon in their root systems and making the entire system carbon negative? Coupled with that, if we can look at high nitrogen uptake, which will have the dual purpose of enhancing nutrient management and protecting our water quality, while also providing a high quality feed for sheep and goats, we get additional ecosystem services and value for the farmer. So those are some of our goals in our long-term breeding program. We work with many different species of willow, and many of them can hybridize. So we have focused on topics of hybrid vigor. Many of our species have different ploidy levels, so we actually make crosses between tetraploid and diploid species and have done a lot of work to characterize heterosis in triploid species hybrids. And we do see consistent heterosis in our triploids, which also have the advantage of being sterile, which reduces their potential for invasiveness. We have used multiple different trait mapping approaches to study willow genetics and to identify markers for marker-assisted selection. Although to date we have not been using a lot of marker-assisted selection in our willow program, we are very close to identifying some of the major genes that will be important for long-term genetic improvement through these different mapping approaches. So we have used association mapping, or GWAS. We have generated an F2 mapping population of Salix purpurea, and that is our reference species. We have worked with the Department of Energy Joint Genome Institute to develop very high quality PacBio genome assemblies of a male and female willow I should have mentioned willow is dioecious, so making these crosses involves crossing between a male and a female sibling in a population. We have also most recently developed a series of F1 species hybrid mapping populations by crossing different diploid species relatives to our reference species Salix purpurea. And we believe that will be very fruitful in the long run for intergressing unique alleles from these other diverse species. We're particularly interested in resistance to rust. Rust is our major pathogen threat to willow, and some of these related species to Salix purpurea bring resistance to our common rust species here in the United States. So this is the latest trial that we have planted this past spring for genetic mapping. It includes about 425 individuals in our S2 mapping population derived from our monoecious individual, as well as another about 180 diverse unrelated individuals for GWAS mapping or association mapping. We plant in 3 plant plots in a complete randomized block design in single rows, so it's relatively easy to cut back with our tractor mounted single row harvester. And again, the goal would be to identify genes for rust resistance, for resistance to insect pests, and to identify variation in other morphological traits which will lead to yield increases. So you're probably not accustomed to growing willow as an agricultural crop. What you see behind me are plants that are in their second growing season after cutback. So this is a short rotation coppice system where we originally plant from cuttings. So our breeding program is aimed at producing clonal cultivars propagated through stem cuttings, which we produce in nursery beds. Those cuttings can be planted mechanically with a planter that will take long whips and cut on the fly into 8-inch cuttings, which it plants in the ground. We typically will plant in a double row spacing, 30 inches between those rows, 2 feet between plants, which has been optimized for harvesting with a self-propelled forage harvester with large saw blades on the front. So after original establishment by cuttings, we typically will cut back the field or coppice it. That stimulates rapid regrowth of multiple small diameter stems in year 2, and then we will typically allow those plants to grow 3 or 4 years before we come in with the forage harvester to cut them back down to the ground as low as we can get, producing wood chips that again we deliver to a power plant or to a conversion facility. The plants will vigorously resprout the following spring after that harvest, and we can continue and sustain that cycle of 3 to 4 year harvest cycles for at least 7 to 10 times, meaning that the crop is sustained and continues to sequester carbon for 25 to 30 years. So these willow crops need to have durable disease resistance for more than 25 or 30 years, making that one of our most urgent breeding needs is to identify primarily rust resistance, but also resistance to insect pests, which will become more persistent with more life cycles as temperatures begin to increase through climate change. Finally, we have been looking at sex determination and the mechanism of the sex chromosome in Salix, especially as it relates to poplar and the evolution of sex determination in the Salicaceae. Very interestingly, there are different mechanisms for sex determination across the Salicaceae, either XY or ZW, ZW being where the female is the heterogametic sex, as it is in birds and reptiles. So we have flip-flopping of the sex determination system across the Salicaceae, and also movement of the sex determination region from different chromosomes. In the species that we work with, the sex determination is on chromosome 15, but in other species of Salix, it has moved to chromosome 7, and in poplar, it can be found on chromosome 19. So those are interesting topics of what is the evolutionary driver for dioecious in the Salicaceae, and what is the driver for ZW versus XY, and the movement of the sex determination region across different chromosomes. Is the sex determination region linked to genes that are driving sex dimorphism, for example? So we've been doing a lot of research looking at sex dimorphic traits most recently. The final tool that we have to address that question is we do find monoecious varieties of willow. So individuals that produce both male and female flowers on a single individual. We have used that to create an S1 and now an S2 mapping population, which we have genotyped and have recently planted here for phenotyping. So that will give us yet another mapping tool to identify important traits in Salix purpurea in the S2. And we also expect to be able to identify loci that are responsible for inbreeding depression in that population.
This has been a production of Cornell University, on the web at cornell.edu.